My key task is really to help you make what is a very important decision, and that is what to study at university. Clearly, I hope that some of you will choose to study the Biomedical Sciences at King's, but if some of you choose to study the Biomedical Sciences and go somewhere else, I will also feel I've been successful in my task, because making that decision about what interests you and what you would like to study for 3, 4, potentially 5 years is critically important. The Biomedical Sciences is the science basis that underpins medicine. And the reason we offer degree programs in the Biomedical Sciences is because we have, alongside the Biomedical Sciences, we teach medics, dentists, pharmacists, physiotherapists, dietitians, etc. And all of those people need to be taught the science of their clinical disciplines. But many of us, myself included, like the science, but we're not that bothered on the clinical aspects. And you can therefore do degrees in the sciences, in the Biomedical Sciences, and it's a very worthy thing to study at university. So you're going to be taught your Biomedical Sciences alongside all these other people, and taken together, we're one of Europe's largest centres for medical and professional healthcare education. And the critical thing we do is alongside the Biomedical Sciences, we're looking to make sure that the discoveries we make whilst we're carrying out research in the Biomedical Sciences, some of the research that you will hear about as part of your degree programs, because as a research-intensive university, all your lecturers will be carrying out research. So some of the research that we do, some of the research that you will get involved in as a student, that research has got to be ultimately of benefit to society, of benefit to patients. So the kind of journey from bench to bedside, which you'll see in a second, is critically important to us. The other key thing I want you to think about as we go through the talk is that you may not know at this stage which particular biomedical science you want to study. There's a great deal of overlap between them. And the important thing I want to emphasize is this word flexible. As we'll see towards the end, you have the opportunity to change between these programs once you get here. So don't worry too much about whether you know you want to be a biochemist or an anatomist. You have the opportunity to change once you get here. And ultimately, we think we do good teaching, we do good research, our students are highly thought after, and towards the end, I'll just give you some examples of the kind of things our graduates have gone on to do once they leave King's, because they do go on and do fantastic things. So rather than me just going through a list of the Biomedical Sciences, I'm going to use this scenario, it's a hypothetical scenario, to try to show you where biomedical scientists might be involved. And this hypothetical scenario involves this student, Keisha, 22-year-old student, come to university, and to cut a long story short, Keisha has developed schizophrenia. It's quite typical that students at university develop schizophrenia because it's a condition that actually develops in late childhood, early adulthood. So quite often the first symptoms of someone developing schizophrenia occur whilst they're at university. The good news is there are ways we can treat schizophrenia very effectively, and one of the ways we can treat it is using drugs, and this drug here, olanzapine, is a very effective antipsychotic drug. But like all drugs, there is no drug that we give patients today that is absolutely safe. All drugs have side effects, and olanzapine has a rather peculiar side effect. Olanzapine owes its ability to treat schizophrenia because it interferes with a neurotransmitter in the brain called dopamine. And in patients with schizophrenia, the levels of dopamine in the brain are slightly lower than normal. But that neurotransmitter dopamine is also involved in what we call the reward pathway. And the reward pathway is the thing that makes some critical things that we do pleasurable. Reproduction, eating, in some cases taking recreational drugs. Because that reward pathway is very highly developed. And what happens is when you do something that's rewarding, you trigger off the release of dopamine in certain parts of the brain in this reward pathway. And what we know is that olanzapine, because it interferes with dopamine, it can make the experience of eating far more rewarding than it usually is. And as a consequence, typically patients on olanzapine often will start to gain weight because they eat more than they used to because they find it very rewarding to eat. To the extent that we typically will actually give patients who are given olanzapine dietary advice. They'll be given a dietitian to help them balance their diet to try to stop them gaining weight. But Keisha hasn't been fortunate, and she has continued to gain weight to the extent that she has developed diabetes. So think what we're saying here, we've got an individual who has developed diabetes as a consequence of the drug she is taking to treat her schizophrenia. And we know she's developed diabetes because her blood glucose levels are higher than they should be. They should be about 4 to 8 millimoles per litre. She's got ketones and glucose in her urine. And As a result, Keisha is rushed to hospital, and the question is who will be involved in Keisha's case? Well, there are lots of clinicians who will be involved in Keisha's case: doctors, pharmacists, nurses, dietitians, etc. I'm going to be talking about these ones down here at the bottom, the research scientists. And just to emphasize how multidisciplinary we are in terms of the people we treat, people we teach here at King's, all of these folk, with the exception of optometrists and podiatrists, all of these folk are taught in our Faculty of Life Sciences, our health faculties across the university. But I'm going to be focusing on these ones at the bottom, the research scientists, because they are the people who will start by doing a degree in the biomedical sciences. And these are the biomedical sciences. I'm not going to go through them in detail, but as I said, it's the science base that underpins medicine. Now, what I'm going to do is I'm going to start small and get big. So I'm going to start small by looking at the kind of molecular level and think about biochemistry, sometimes also called biological chemistry. And everything we do, whether we like it or not, everything we do is governed by chemical interactions occurring between cells and within cells of our body. We are just great big bags of chemicals. And understanding how those different chemicals interact with each other to control fundamental life properties is the business of a biochemist. Now, in this case, you don't need to know the details of this, but this is the bloodstream, this is a membrane of a cell, and this is inside the cell. And this blob here is insulin. And as you know, in patients with diabetes, the problem is that either they're not producing enough insulin, or the insulin they do produce is not effective at activating different cell types in the body, so they become insulin resistant. And in the case of Keisha, it's probably insulin resistance that she's developed. And the reason I put this up is this is insulin, and what it does is it acts on what we call insulin receptors in the cell membrane to trigger off a hugely complex biochemical cascade inside cells. Each one of those multicolored blobs is a different chemical, a different enzyme, a different protein that's altered as a result of insulin acting on this cell. And really what's remarkable is that it doesn't go wrong more often, because all of these chemical interactions have got to work in a certain sequence at a certain point in time to really drive that cell's response. Now, in some cases, that will trigger a cell to divide. For example, it will trigger off cell division. And in that case, in many of the kind of types of cancer treatment that we're looking at now, where cells are dividing inappropriately, we're starting to target with drugs these biochemical cascades inside cells. So understanding how these cascades work, understanding what happens when they go wrong, and thinking about how we might interfere with them using drugs, for example, is what biochemists get up to. So if you like chemistry but would like to target your chemistry to kind of biological problems, then this kind of program may be a good one for you. Now, staying small, genetics. Genetics, how the traits and how our traits and characteristics are passed from one generation to another in our genes. Now, some forms of diabetes have got a genetic component. Schizophrenia has got a genetic component. We know that it's got a genetic component, but there is no one schizophrenia gene, for example. There are probably more than 10 different genes, and if individuals have mutations in those 10 genes and they all come together, then it may be what leads to schizophrenia. So there is certainly a genetic component, but how strong that genetic component is yet to be determined. So a geneticist may be looking at how the genetic background of an individual makes them more susceptible to disease. Another alternative may be looking at how they may respond to drugs. Pharmaceutical companies are very interested in what's called personalized medicine. And personalized medicine is where in 5, 10, 15, 20 years' time, you will go to your doctor will take a blood test, and they will look at your gene profile based on that blood test. And from that gene profile, they will decide which drug to give you to treat your disease. It may be a drug that we know will work better in you because of your genetic background. It may be a drug that won't cause a particular side effect in you because of your genetic background. If you think about what we do at the moment, when we develop drugs which are effective in all the population. If I was to try to develop a new anti-schizophrenic drug and it caused a really life-threatening side effect in 10% of the population, it would not get onto the market. 90% would be okay, but if 10% had that side effect, it would not go onto the market. Pharmaceutical companies like the idea of personalized medicine because they would be able to identify from the genetic profile those 10% and say you will never get the drug. But there's a bit of an ethical dimension to that, because what happens if you're in that 10%? Pharmaceutical companies are very good at developing drugs which will have a big market, 90%, but they may not be keen to develop a drug for only 10% of the patient population who have a particular condition. So alongside the kind of genetics of disease and genetics of your response to drugs, we do talk a lot about the kind of ethics surrounding the information we're getting from the Human Genome Project and how we have to think about that before we take it forward. Pulling back in my kind of metaphorical microscope away from the molecular level, anatomy, developmental and human biology. Now, you may be saying, why do we need to know anything about anatomy? You know, anatomy is an old subject. Anatomy is understanding how human evolution, the structure of organs influences function. Why is the heart heart-shaped? To make it an effective pump. Now you say, well, what's new in anatomy? Da Vinci was drawing anatomical drawings decades ago, centuries ago. Well, I'll give you an example. This is the pancreas, and as you know, the pancreas is the organ that secretes insulin. And a patient with diabetes, they're not secreting enough insulin. Now, one way that we are thinking about treating the main form of diabetes, so-called type 1 diabetes, is to replace the pancreatic beta cells in the pancreas, which are not functional, using stem cells. You've heard of stem cells. Stem cells have this unique ability to change into any cell type in the body. And we know that in a test tube, you can take stem cells and treat them with our various chemicals in a certain combination and at certain time points to divert them, to differentiate them into pancreatic beta cells. And in theory, we could take stem cells, transplant them into the pancreas, and trigger them to become pancreatic beta cells and therefore replace the damaged beta cells that are in the pancreas that are no longer secreting insulin. So it's kind of transplant approach. And that is fantastic. Developmental biologists have told us what we need to do to the stem cells to make them pancreatic beta cells. We've just got to get them into the pancreas. That's where the anatomy comes in, because where do you put them in the pancreas? Because the pancreas has got two roles. The endocrine pancreas secretes insulin into the bloodstream, but another part of the pancreas, the so-called exocrine pancreas, secretes digestive enzymes into the bile duct and eventually into the gut, where it's involved in digestion of food. And clearly, you need to put the pancreatic beta cells into a part of the pancreas that's exposed to the bloodstream so that they secrete insulin into the bloodstream, as opposed to secreting insulin into the bile duct and into the gut, where it would have no effect whatsoever. So understanding the structure of the pancreas is critically important in understanding how we can start to approach those types of treatments, for example. So the developmental biologists tell us something about stem cells and how to treat them to become pancreatic beta cells, and the anatomists tell us something about where to put those pancreatic beta cells if they're to become effective in replacing the deficient cells. Medical physiology. So physiology is how does the body operate under normal, healthy conditions? What is in the main systems of the body, the cardiovascular system, the respiratory system, etc. And just to give you an example of this, the question is, well, why do we have insulin? Well, the reason we have insulin is because we need to maintain a constant supply of blood glucose. And the reason we need to have a constant supply of blood glucose is this thing, because your brain is incredibly greedy. It is incredibly greedy for energy. The hundreds of billions of nerve cells in your brain are firing off electrical impulses, even if you can feel your eyelids starting to droop at this moment because it's a hot day and you're listening to a really dull talk on Biomedical Sciences. Your brain is firing off electrical activity, and that uses up a lot of energy. And your brain needs a constant supply of glucose to maintain that. You probably felt that feeling of hypoglycemia when you start to feel a little bit shaky, you start to feel a little bit faint. That's because your brain is running out of the energy supply of glucose, and you've got to have a banana or something to get you back. But this is a problem for the body, because when you eat, your blood glucose concentration goes up. Then between meals, your blood glucose concentration goes down. Then your next meal, it goes up. Then between meals, it goes down, etc. So your brain needs this constant supply of glucose, but what it gets through your eating is a waveform of glucose, up and down and up and down. And so what the body then does is use insulin, because insulin is released from the pancreatic beta cells when you've got a high blood glucose concentration, just after you've eaten. And it acts on all those cells, fat cells, liver cells, muscle cells, on those receptors to trigger off those biochemical cascades to make those cells take up glucose and store that energy. So in times of plenty, just after your meal, insulin's acting to make these cells store energy. And as time goes on and your blood glucose concentration falls, insulin stops being secreted. These cells then start releasing that glucose back into the bloodstream to maintain that constant supply. And when insulin goes wrong, we lose this ability to control our blood glucose levels. And physiologists tell us about that scheme, and they tell us, for example, how the best way to treat patients by giving them insulin. When do you give them insulin? How often do you give them insulin, etc. So that's what physiologists do, and there's still lots to find out about the normal or the healthy workings of the human body, and more related to that, what we call pathophysiology, is what changes in disease, for example. How does the cardiovascular system change in someone who suffers from high blood pressure? Immunology. Immune system's good. The immune system protects us against infection. It protects us against invading pathogens. But increasingly we know that many diseases have an autoimmune component. And we now know that the main form of diabetes, type 1 diabetes, is an autoimmune disease. I mentioned it's caused by the fact that the pancreatic beta cells are no longer functional in the pancreas. And the reason they're no longer functional in the pancreas is because your immune system, or the individual's immune system, has destroyed them. The immune system has, for whatever reason, misrecognized the individual's pancreatic beta cells as being foreign bodies, and it has attacked them and it has destroyed them in the same way that it would attack a bacterial infection or something, so-called autoimmune disease. So understanding why the immune system has made that mistake and has started to attack the beta cells is an important part of what immunology is about. And there are many diseases ranging from Alzheimer's disease, rheumatoid arthritis, which have an autoimmune component. But equally, if we now know that the immune system can target some of the cells in our body, if we can understand what's making it do that, there is an alternative way to think about that, and that's coming to when it comes to cancer treatment. In other words, can we make the immune system recognize cancer cells and start to attack them, start to destroy them, or at least deliver drugs to those cancer cells? So is there a way that we can use this ability of the immune system to have an autoimmune target to treat cancer, for example, and minimize the likelihood of the fact that at the moment when we give patients chemotherapeutic drugs to treat cancer, we cannot get a drug that will only harm the cancer cells, it will also cause harm to other cells in the body, which leads to the side effects that we know of from cancer treatment. So understanding immunology is important for disease, but it's equally becoming increasingly important as to how we might treat some disease using this ability of the immune system to recognize cell types. Neuroscience, the nervous system, the brain, hundreds of billions of neurons in your brain. Obviously in this case, in the case of Keisha, we might think about what has changed in the brain of Keisha to lead her to develop schizophrenia. And the answer is we don't really know. We know a lot about individual nerve cells. So we know a lot about how individual nerve cells regulate their electrical activity. And if you do choose to come to King's and you get a lecture from me, I spent my PhD looking at the effects of morphine in a particular type of nerve cell in your brainstem called the locus coeruleus. And I will put you in a corner and you will be begging for mercy as I keep telling you about all the wonderful things that happen there. But how you go from the effects of drugs or the effects of individual nerve cells and amplify that up to the hundreds of billions of cells that come together to produce powerful emotions like love, hate, anxiety, depression, we just don't know. We're really in the foothills of understanding that. That is the business of neuroscientists, trying to understand the brain and how it works. And it goes from the cellular aspects right up to the kind of behavioral aspects, kind of psychology type approaches. Now, one of the reasons that I'm a pharmacologist, and so I'm interested in drugs and how we develop drugs. And one of the reasons that neuroscience has proved quite a tricky aspect to treat with drugs, for example, is because quite often these are very human conditions. You might not like it, but if you will regulate your blood pressure in much the same way that a mouse or a rat regulates its blood pressure. And so if I was setting out to develop a new drug to treat high blood pressure, I can make a pretty reasonable guess that if it lowers the blood pressure in a mouse, it will probably lower the blood pressure in a human. But that doesn't work with psychiatric disorders. I wouldn't know, I wouldn't recognize, I don't even know if mice suffer from depression. I don't know if they suffer from anxiety. So these are very human conditions that are kind of typical ways that we used to develop drugs are not really effective in. So one of the great unmet pharmacological challenges is how we treat some of these disease states of or psychiatric disorders of the brain. Which leads me to pharmacology, because pharmacology pulls all those different aspects together and puts the drug and the medicine at the center of what we do. So how can we use drugs? By understanding the physiology and understanding the pathophysiology, what those changes are, how can we use drugs to treat that disease state? And in many cases, we are very effective at using drugs. But important thing to remember is in many cases, we do not cure the disease. Think about high blood pressure. We know high blood pressure is important to treat because ultimately it will put an excessive strain on the heart. So we know that if an individual suffers from high blood pressure, it's important to treat them with drugs. But the important thing to remember is we do not cure high blood pressure. We give the patient a drug and they will be on that drug for the rest of their life. Take the drug away, their blood pressure goes back up. We haven't cured the disease, it's still there. We don't understand what has changed, the disease is still there. So in many cases, we're taking drugs to provide what's called symptomatic relief. We're relieving the symptoms, but we don't cure the disease. Now, this is a drug called glibenclamide, which is used to treat diabetes, the type of diabetes that Keisha is suffering from. And glibenclamide's got a relatively simple mechanism of action. This is its molecular target. It's actually a huge protein molecule. You can't see it, but there's actually 8 different proteins come together in this large molecule. And this large molecule is inserted into the lipid, the membrane that surrounds the pancreatic beta cell, the cell that should be secreting insulin. But in Keisha's case, it's not doing it. You're looking down on top of this large thing. It's what's called a potassium ion channel. That's a potassium ion, a positively charged potassium ion, and it flows all the way through the membrane. So this potassium ion goes through this channel from outside the cell inside. And what you have to do in a pancreatic beta cell to make it secrete insulin in response to glucose, remember what's happening, glucose goes up, the pancreatic beta cell secretes insulin. By triggering off a biochemical cascade inside the pancreatic beta cell, what glucose does is it closes that ion channel. There's a gate, clams shut, such that you would no longer be able to see that hole through which the potassium ion can flow. It closes the ion channel. Now, because potassium ions are charged and they're flowing across the cell membrane, that's an electrical current, a tiny little electrical current, but it's there nonetheless, flowing across the cell membrane. And switching off that electrical current is a stimulus for the pancreatic beta cell to secrete insulin. And in the case of Keisha, for reasons, well, we do understand, but for reasons I don't have time to go into, this potassium ion channel is no longer closing in response to insulin, in response to glucose. And what this drug does, it comes along and it tries to go through this ion channel. It sticks its head in the ion channel and blocks it. So effectively, the drug is acting like the gate for the ion channel. It's just simply sticking its head in there, trying to flow through the ion channel, gets stuck, and closes off the potassium current. And that will make the pancreatic beta cell, in Keisha's case, secrete insulin. So pharmacologists spend a lot of time looking at the structure of drugs, the structure of targets, and trying to get them to work together. Right, I mentioned flexibility. So you've now heard about most of the biomedical sciences, and the important word I want to take away is this idea of flexibility. These are our degree programs, and I hope that those of your interest will be thinking at one of them up there. The important thing to remember is all of these programs follow the same first-year curriculum. They all follow the same first-year curriculum. You do introductory modules in all the biomedical sciences. The entry requirements are the same. So even if you think at this moment you want to be a biochemist, at the end of your first year, I will have converted you to pharmacology. Don't worry too much if you don't know which of these it is you want to study. Get here, as I say, the entry qualifications, make your best guess, and then once you're here, you've got the opportunity to change at the end of your first year or sometimes at the end of your second year. One particular program that's worth noting is what's called the BSc in Biomedical Sciences. If you want to be a pharmacologist, we tell you a number of core and compulsory modules you must take. There are optional modules as well. But as a biomedical scientist doing a BSc in Biomedical Sciences, you can pick and choose whatever modules you want to put them together to a program of study that suits you. You get a course advisor to help you with that choice, but there's a huge amount of flexibility in there. So you start off with this common year 1, and then by the end of, it comes to years 2 and 3, you start to kind of focus in on what interests you. And the critical thing is what interests you. because it's really easy to study at university and get a good degree if you're sitting in the library reading about something you're interested in. It's that bit harder if you think, oh, this is a bit of a chore having to sit down and read another book about pharmacology or whatever. You'll have seen these Mside degree programs. Just to mention, they are 4-year degree programs, and they're really focused on those who at this stage know they want to go on to do a research career. So there's a 4th year after the typical 3-year BSc where you stay at university and do an extended research project. So if at this stage you know that research is where you would like to get to, you'd like to do a PhD and work in a place like this or work in the pharmaceutical industry, think about whether registering for an MSci or applying for an MSci program at this stage would be good. But again, even if you don't know this at this stage, if you come for a BSc, you can switch to that program once you've been here for a couple of years or so. So the flexibility is there. There are lots of things you can do to enhance your degree, one of which is leave King's. You can go into one of our international partners, for the Karolinska Institute, for example, National University of Singapore. Students will typically spend their 2nd year at NUS or Karolinska and then come back to us for their final year. You can do an industrial year, what we call an extramural year. You can do research projects in our labs, etc., etc. So think about the things you can build around your degree program that just make it that bit more of a better experience for you. And our students go on and do fantastic things. You know, these are kind of lab-based careers. I tend to think about careers in 3 different lots. Those who wear a lab coat, those who wear the white coat, so people who work in labs, the kind of traditional careers for students who do biomedical degrees. And these are kind of lab-based degrees in different places, including universities and National Health Service. But there are also BSc students who get a degree in the biomedical sciences but don't want to wear the white coat. They like their science, but they want to use their science in different ways. And they might go into, for example, science policy. They may go into science journalism, work for the BBC, regulatory affairs. Fantastic career opportunity. So you can use your science degree without actually working in a laboratory. And finally, there are students who go on and make more money than I could ever dream of. They cross over London Bridge, go into the city. But what they're doing is they're taking the skills they have learned during a science degree to use them to enter the graduate market. If you think what a scientist does, a scientist identifies a problem. They develop a hypothesis to explain that problem. They then carry out experiments to test that hypothesis. Depending on the results of those experiments, they then change the hypothesis. And everything you do in life is a series of problem-solving initiatives. And that scientific way of solving problems, of hypothesis testing, will set you up for whatever career. Alongside things like communication skills that you will be taught, you will learn how to stand on your hind legs, give talks to different audiences, etc. And then, or you may decide to be a student and go on to do PhDs, master's programs, you want to get into graduate entry into medicine or dentistry. So lots of career opportunities open to you. Thank you.